Swati, yes.
I, Swati Jain, National Coordinator, Women's Forum SPSR, on behalf of SPSR, welcome you all to attend a national webinar on the topic NDMA impurity in medicine, a source of bewilderment for the patients. Let me extend a hand of gratefulness to highly learned Professor Dr. Shashi Alok, President SPSR, to have given me an opportunity to transact such an informative webinar on an exclusive topic today. Welcome, Dr. Shashi. I would also like to welcome our eminent keynote speaker, Dr. Geer Mohammad Ishaq, sir, Associate Professor at Department of Pharmaceutical Sciences and Coordinator at Human Resource Development Center at University of Kashmir. SPSR feels highly honored to have got an opportunity to share a part of your vast knowledge today, sir. I also welcome highly knowledgeable Dr. Abhishek Mathur, scientist, consultant, and techno-commercial expert, and an executive director technical at NCS Green Earth Private Limited in Nagpur, who has gratified us by accepting to be a part of this webinar today. I also heartily welcome the entire team of SPSR, Secretary SPSR, Mrs. Monica Sabbarwal, Joint Secretary SPSR, Mr. Saurabh Kose, President Women's Forum SPSR, Dr. Padmini Shukla, and all other members who are diligently working to strengthen the foundation of SPSR and right now attending this significant webinar today. Since this webinar is organized by SPSR, I would like to elucidate its purpose. Society of Pharmaceutical Sciences and Research is an educational, non-profit organization. It is committed to encourage the study and practice of pharmacy education and research. The mission of SPSR is to promote the pharmaceutical sciences in all aspects. Our vision is to promote and advance the science of pharmacy and its applications in India. We look forward to promoting and facilitating the acquisition, dissemination, and exchange of pharmacy knowledge and information about the practice of pharmacy and pharmaceutical services. Now I request Dr. Shashi Alok to kindly take the lead. Over to you, sir. Over to you, Shashi sir.
I think he's not available now.
Yeah. I guess there is some net connectivity. I would request Dr. Mathur to kindly do the proceedings.
Swati, thank you, Swati Jain. Okay. Thank you, Swati man. A very good morning to all. I am myself Dr. Shashi Alok, President SPSR. On the behalf of Society of Pharmaceutical Sciences and Research, I welcome Honorable Speaker Dr. Geer Mohammad Ishaq, Associate Professor, Department of Pharmaceutical Sciences and Coordinator Human Resource Development Center, University of Kashmir, Jammu and Kashmir. I also welcome all the SPSR office bearer, and all the participants, national as well as international, who are presented here in this SPSR National Webinar. So SPSR today going to organize national webinar of NDMA impurities in medicine. So I again welcome Honorable Speaker Dr. Geer Mohammad Ishaq, and thank you, sir, for accepting our invitation for this national webinar. And now I would like to request to Dr. Abhishek Mathur kindly introduce today's eminent speaker. Over to Dr. Abhishek Mathur.
Thank you, Shashi sir. It's my great privilege to introduce Dr. Geer Mohammad Ishaq sir, who is an eminent professor in Department of Pharmaceutical Sciences, University of Kashmir, Jammu and Kashmir, India. He is today's speaker, and he's been working for the last 18 years and is an instrumental in designing and starting a postgraduate program in pharmacy practice at the University of Kashmir, and is presently in charge of this postgraduate program offered by the University of Kashmir. Dr. Geer Mohammad Saab has also served as a drug control officer with the government of J&K for a period of about 3 years prior to joining the Kashmir University. He holds a doctorate in pharmacology and has research interest in pharmacovigilance, pharmacy practice, pharmaceutical care, drug utilization, evaluation, pharmaceutical policy, quality control, and rational use of medicines. Dr. Geer has so far published 48 research papers, 46 abstracts, and more than 100 popular articles, and presented about 42 papers in various national and international conferences of high repute. His team has won 5 Best Paper Awards in various conferences and journals. He has delivered more than 100 extension lectures on various topics outside his department, besides reviewing numerous papers for various national and international journals of repute. He has reviewed and revised the drug policy in his own home state, J&K, and now he is also dispensing his additional duties as a coordinator, Human Resource Department Center, University of Kashmir. And in his capacity, he has organized 19 teacher training courses as course coordinator. Dr. Geer Mohammad Saab has also been appointed as a member expert committee on antibiotic utilization under National Drug Utilization Evaluation Program of India by the Indian Council of Medical Research, ICMR, New Delhi. He has also served as a country leader uh, in BRICS Medicines Alliance and as a Joint Secretary, North Zone Association of Pharmaceutical Teachers of India, APTI. Recently, Dr. Geer Saab has also served as a Deputy Secretary National Webinar in Jammu and Kashmir. So I wholeheartedly welcome Dr. Geer Mohammad Saab on behalf of SPSR. Now over to Dr. Swati Jain.
Thank you so much, Dr. Mathur. Sir's profound personality itself speaks volumes about the expertise he has, and that is the reason we have got over 1700 registrations today, not only from India, but from different parts of the world, like Egypt, Nigeria, Kingdom of Bahrain, Bhutan, Pakistan, Saudi Arabia, Tanzania, Australia, Nepal, Azerbaijan, Syria, Algeria, Philippines, UAE, Germany, Macau, South Korea, Jordan, Iraq, Taiwan, Indonesia, Yemen, Ethiopia, and others. So, viewers, regarding today's webinar topic, nitrosodimethylamine or NDMA is an impurity which is commonly found in water and food, including grilled and cured meats, dairy products, and vegetables. Medicines and regulatory authorities first became aware of the presence of nitrosamine impurity NDMA in products containing valsartan in July 2018, which resulted in voluntary product recall. The unexpected discovery of these impurities has subsequently fueled the development of new manufacturing requirements for these medications, and pharmaceutical companies have had to take precautionary measures to mitigate the risk of producing nitrosamines during drug synthesis. These impurities have been shown to be probable human carcinogens and others deemed to be a health concern. SPSR feels privileged to get a chance to know about this topic through learned speaker Dr. Geer Mohammad Ishaq sir with insightful knowledge in this area. Over to you, sir.
Thank you very much, Dr. Shashi Alokji, President, Society of Pharmaceutical Sciences and Research, Secretary, SPSR, Mrs. Monica Sabarwal, Joint Secretary, Dr. Saurabh Kose, Dr. Abhishek Mathur, and Mrs. Swati Jain, plus all other members and office bearers of the Society of Pharmaceutical Sciences and Research, and all the participants of this webinar attending from within and outside India. First of all, I wholeheartedly thank you all for inviting me and giving me this opportunity to address to your forum, to your members, and to your audience on this vital topic of NDMA impurities in medicines. It's a great privilege and honor for me to be addressing this learned audience today on this particular topic. I'm highly thankful to all of you for sparing time and giving me this opportunity and inviting me on this particular topic. Thanks a lot. Now, so far as today's topic is concerned, and it is equally heartening to know that people are attending this webinar from all across the world, from so many countries, as Dr. Swati Jain has just now enumerated. So, I would be looking forward to a very interesting and informative session today. So far as NDMA impurities are concerned, as very rightly and pertinently briefed by Mrs. Swati Jain, that it all started in July 2018, when US FDA made the revelation that this particular impurity has surfaced in valsartan, the angiotensin receptor blocker, and then it was followed by many other drugs, which I will be detailing out. So in today's webinar, we will be discussing how this all issue surfaced, when were the first reports which came out, then how many drug recalls were made, what were the drugs affected, what were the various companies and their products affected, where is the source of this impurity, origin of this impurity, how this contamination has occurred. And then we'll also look at the various measures which patients need to take, which companies need to take to overcome this problem, which the drug regulatory authorities need to take, and how best they can detect it, and how best they can report it, and how best they can curb it, so that there is minimal damage, minimal hazard caused to the patients. So that is the brief outline of my presentation. So now I would share my presentation so that we can directly start with the presentation today. All right, Ms. Swati Jan, is the presentation visible?
Yes, sir, it is visible, sir.
Yes, sir, it's visible.
So dear participants, dear listeners, the topic, once again, I would repeat, is NDMA impurity in medicines, which is a source of great bewilderment for the patients all across the world. NDMA stands for N-nitrosodimethylamine impurity. It is only one of the types of impurities in the category of N-nitrosamines. So I'll be talking about the other types of impurities in this class. Now, first, this N-nitrosodimethylamine, this was first detected by US FDA in July 2018 in an antihypertensive drug called valsartan, which is an angiotensin receptor blocker. And then subsequently, it was found in other antihypertensive medicines like amlodipine, losartan, irbesartan, hydrochlorothiazide. Then it was also found in gastric acid prevention drugs like ranitidine, nizatidine, and very recently, it has also been observed and detected in anti-diabetic drug called metformin, which is the first-line therapy for diabetes patients. In valsartan, this impurity was detected in July 2018. That was followed by candesartan. In October 2018, it was detected in irbesartan, followed by olmesartan. It was detected in November 2018 in the drug losartan, and then in this anti-peptic ulcer drug ranitidine in September 2019, followed by nizatidine in January 2020. And this year in July 2020, this particular contamination has again surfaced in a very widely used anti-diabetic drug, metformin, which is the first-line therapy for type 2 diabetes mellitus. And as a result of all these popular drugs getting affected because of this impurity, a lot of confusion, chaos, and a lot of bewilderment and anxiety has surfaced among the patients all across the world, because patients really don't know whether they should continue with the use of these drugs or not, and what exactly they should do. It was the European Medicines Agency which first detected this particular contamination or impurity in valsartan on July 5th, followed by US FDA on July 16th, 2018. And the drug was belonging to the, it was originating from Chinese manufacturing firm Zhejiang Huahai Pharmaceutical Company. And since then, as many as 53 drug recall notices have been issued by US FDA. And 16 of these drug recall notices have been issued this year in 2020 alone. And till September 2019, as many as 2,053 drug batches have been recalled. And now I think it might have touched around 3,000 by now. So 3,000 batches have been recalled from the world markets, particularly from the US markets by the US FDA, since this contamination has been detected. As I said in the beginning itself, N-nitrosodimethylamine is not the only impurity of this kind. There are many other types of nitrosamine impurities which have a common moiety, and these impurities include N-nitrosodimethylamine, which is mostly found in the medicines, N-nitrosodiethylamine, which is mostly found in the food products, vegetables, etc. Then we have many others like nitrosomethylethylamine, nitrosodi-N-propylamine, nitrosodi-isopropylamine, N-nitroso-N-butylamine, N-nitrosodiphenylamine, N-nitrosopyrolidine, N-nitrosopyperidine, and N-nitrosomorpholine. So just by looking at their chemical structures, if you have studied pharmaceutical chemistry, you can easily name these impurities by following the IUPAC nomenclature. But one thing common in all of them is that, mostly amines are affected by this impurity. It's not an impurity or contamination which has been deliberately incorporated by someone. It is an impurity which has somehow creeped into the manufacturing process. And mostly the amines are affected by this contamination, either primary amines which have 1-methyl substitution, or secondary amines which have 2-methyl substitution, or tertiary amines which have 3-methyl substitutions on the nitrogen atom, or quaternary ammonium which have 4-methyl substitutions on the nitrogen. So it is, the precursor is either the dimethylamine or trimethylamine or dimethylaniline. These are the precursors on which nitrosation occurs. Nitroso derivative is added to these precursors. Upon nitrosation, they lead to the formation of either N-nitrosodimethylamine or N-nitrosodiethylamine. So that's how this particular nitrosodimethylamine derivative impurity is getting incorporated into the manufacturing process. Now, so far as the foods are concerned, nitrosamines have been formed by reaction of nitrogen oxide with amines. As you can see in this slide, nitrosamine impurities, it was first detected way back in 1950. It's not something which is being detected now. Nitrosamine was found to be carcinogenic way back in 1950s. And since then, it was found in many foods. It is present in water, it's present in air, it's present in food substances, it's present in cosmetics, and in food substances, but in particular, it is incorporated by the reactions of nitrogen oxide with amines. So nitrite present in the food, either obtained from the fertilizers which we use, or obtained from the preservatives which we use to preserve the food substances. This nitrite is hydrogenated to hydrogen-nitrogen oxide, H2NO2, as you can see in the chemical reaction on the right side. And this happens in acidic conditions, that nitrite is hydrogenated to hydrogen-nitrogen oxide. Now, this hydrogen-nitrogen oxide, H2NO2, reacts with another molecule of nitrite to form nitrogen anhydride after dehydration. That is N2O3. And this nitrogen anhydride donates the nitroso group to the amines in the food to produce N-nitrosamine contamination, to produce N-nitrosamine impurity. And mostly secondary amines are affected the most. Usually tertiary amines are very rarely affected. And primary amines, even if affected, they do not last long because they are not quite stable. So it is the secondary amines which are affected the most. And in spinach, cabbage, carrots, and other vegetables, this nitrate has been reported to be reduced to nitrite by the presence of microorganisms. So this nitrate can be either obtained from the fertilizers or from the preservatives that we use, or it could be reduced to nitrite by the presence of microorganisms. And the nitrosating reaction may also occur in the stomach itself by the reaction of nitric oxide from nitrite or nitrate with amines in acidic conditions. In the acidic conditions, it was observed that even some experiments were carried out that ranitidine may be producing nitrosamine contamination inside the stomach, because inside the stomach there are acidic conditions, and this nitric oxide reaction may be occurring in the stomach itself. So NDMA is the most commonly found impurity among all these nitrosamines in the medicines. NDMA was previously used in the production of liquid rocket fuel, lubricants, sealants, and is also known to be a byproduct in various industries, like in the manufacture of pesticides, rubber tires, alkyl amines, and dyes. This is a very common byproduct in these industries, in these manufacturing processes. And it is believed to be present in many processed foods and alcoholic beverages also, including smoked fish, broiled meat, bacon, cereals, dairy products, some fruits and vegetables. It is believed that, there was one experiment which was carried out that when you broil, in meat, whether broiled meat, baked meat, fried meat, smoked meat, is having more nitrosamine or not, which one is having more. That experiment has also been carried out. So broiled meat was found to be most dangerous because when you broil meat directly on the flames, the fat content present in the meat, it leads to formation of nitrosamines which vaporize, and then they settle back on the meat. And this nitrosamine is carcinogenic, which is known to all. Now, NDMA is one of the most well-characterized, most potent animal carcinogen, and it has been shown to be a potent carcinogen across all species. There is no doubt that nitrosamines or NDMA are carcinogenic. And they get metabolized inside the body by following the same pathway of biotransformation in humans as well as in rodents. They get biotransformed to methyl diazonium ion by the liver microsomal enzymes, cytochrome P450-2E1 enzymes. They get biotransformed to methyl diazonium ion. And this methyl diazonium ion in turn causes mutagenicity, clastogenicity. It leads to the chromosomal aberrations. It leads to the formation of DNA adducts, such as O6-methylguanine, which is another probable carcinogenic agent. So inside the body, this NDMA gets biotransformed to methyl diazonium, which in turn leads to mutagenic challenges, chromosomal aberrations. That is why International Agency for the Research on Cancer, IARC, has classified NDMA as a probable carcinogenic agent to humans and has placed it under Group 2A, that is, which emphasizes that it should be regarded for all practical purposes as if it were carcinogenic to humans, because it is placed under category 2A. Category 1 means definitely carcinogenic, 2A means probably carcinogenic, 2B means possibly carcinogenic, and category 3 means not necessarily carcinogenic. This has been placed in group 2A. Now, how does this impurity or contamination creep into the medicines like these angiotensin receptor blockers or metformin? It is believed that it has been studied well, and it is believed that it could creep in either through the process of manufacturing or through cross-contamination, or because of degradation of the products, or because of the raw material which has been used in the formulations. So it could get incorporated either as the starting material, or at the intermediate level, or as a drug substance or drug product itself. That is, either the starting material itself could be impure. That is what has happened in the case of Zhejiang Huahai company, the Chinese company. The raw material which they supplied for valsartan was itself containing the impurities. And then it can happen in the middle of the process, intermediate product, because of the use of catalysts, reagents, solvents, intermediates, and other byproducts. Any of these could be contaminated. The catalyst, the reagent, the solvent, or intermediates, which is used in the manufacturing of this particular drug. Or it can happen even at the end of the formulation process. That is, the degradation may occur on storage or during transportation, during shipment of the product. So there may be contamination because of high temperature or acidic conditions or any other changes in temperature, humidity, etc. This contamination may creep into the product. Now, when I was telling you which are the drugs that have been affected, if you noticed, I named almost all of the angiotensin receptor blockers, including losartan, valsartan, candesartan, irbesartan, olmesartan. But I did not name telmisartan, I did not name azilsartan, I did not name aprosartan. That is because only those drugs have been found to be getting contaminated by this NDMA or nitrosamine and nitrosodimethylamine contamination. Those drugs which have a tetrazole ring structure, that is which have a 5-membered ring having 4 nitrogen atoms. That is what is meant by a tetrazole ring structure, a 5-membered ring with 4 nitrogen atoms. So as you can see in this slide, valsartan has it in the red color. It is shown 5-membered ring with 4 nitrogen atoms. Candesartan has it, olmesartan has it, irbesartan has it. And similarly, the ranitidine, nizatidine, and metformin also have 4 nitrogen atoms situated adjacent to each other, though not necessarily in a 5-membered ring. But telmisartan does not have it. Aprosartan or azilsartan does not have it. That's why there are least chances of telmisartan, azilsartan, or aprosartan getting contaminated by NDMA. But these drugs are more prone to such contamination because of the presence of tetrazole ring structure. So that is one important characteristic which leads to the propensity of the drug getting contaminated by this impurity. Now, when we look at the origin of this NDMA contamination, it goes back to 2012 to Zhejiang Huahai Pharmaceuticals of China, and it was detected by the European Medicines Agency first on 5th of July 2018. That's when this Zhejiang Huahai Pharmaceuticals, they had made certain changes in their manufacturing process because valsartan was probably coming out of the patent after 20 years. And now they had to look for some other process to manufacture it, or they wanted to reduce waste or improve the product yield. Because of either of these reasons, they tried to change the manufacturing process. And in the process of changing this manufacturing cycle, they made certain changes, as a result of which they made certain changes into the synthetic process of tetrazole ring formation, because of which tetrazole ring formation synthesis changed. And tributylene azide was replaced with a more toxic compound, that is anhydrous sodium azide. Earlier, they were using tributylene azide to generate this tetrazole ring, but now they started using anhydrous sodium azide. This anhydrous sodium azide was used as a solvent apart from dimethyl formamide, and sodium nitrite was subsequently used to quench this excess of sodium azide. Following the synthesis, if there's any excess of sodium azide, that was quenched by sodium nitrite. And in this acidic environment, sodium nitrite forms nitrous acid, which could react with the dimethylamine present in the dimethyl formamide. I told you it is the nitrosation of the amines, carbamates, or amides, which leads to the formation of NDMA. It is the nitrosation of carbamates, alkyl amides, or amides, which leads to formation of NDMA. And this is what was happening here. Tetrazole was a common structural feature among all the angiotensin receptor blockers, which were found to be contaminated. So in the picture, you can see dimethyl formamide leads to the formation of N-dimethylamine, which in turn generates nitrous acid from the sodium nitrite in acidic conditions. And this donates a nitroso group to the amine, leading to the formation of NDMA. Similarly, for N-nitrosodiethylamine, NDEA, which is mostly found in food products, the precursor is the triethylamine, which leads to the formation of N-diethylamine and generates nitrous acid from sodium nitrate in acidic conditions, nitrosating the amine and leading to the formation of NDEA. That's how NDMA is generated. And these are the 5 major types of NDMA impurities with their daily intake limits. These daily intake limits have been fixed by the US FDA. That is, for NDMA, the permissible daily intake is 96 nanograms per day. For NDEA, it is 26.5 nanograms per day. For nitrosomethyl aminobutyric acid, 96 nanograms per day. For nitrosodiisopropylamine, 26.5 nanograms per day. And for nitrosoethyl isopropylamine, 26.5 nanograms per day. Now, these levels are much below than the 190 nanogram dose of NDMA that the WHO would find acceptable. Because in water, I told you, in air and in other cosmetics, it is there. So we consume it somehow, directly or indirectly. And WHO has fixed this limit of 190 nanograms from all the sources. So this limit of 96 nanograms seems to be lesser than that. That's because, elderly people take several medications, 4 or 5 medications, and it's quite possible that they may be taking from multiple sources. Plus, people living in those hazardous environmental conditions wherein there are more chances of accumulating NDMA from the environment. That's why the WHO limit is higher and the limit fixed for the medicines is lower. Now, you will be surprised to know that this particular contamination was also detected first by an online US pharmacy, an online pharmacy of USA, which was named ValueSure. They, at their own level, conducted the tests for the drugs for valsartan and as well as ranitidine. So they found 3 million nanograms per tablet of NDMA levels in ranitidine. Whereas the limit is 96 nanograms, they found 3 million nanograms. And suspecting that the high levels might be because of the drug breaking down to 130 degrees, it is believed that they were questioned that you have used GCMS, that is gas chromatography mass spectroscopy technique, which is a highly abrasive technique which uses high temperatures, and because you have to volatilize the compound. So they were accused that because the temperature, this NDMA might have been generated. So they conducted simulated experiments in which they used ranitidine. They exposed this ranitidine to gastric fluid plus sodium nitrite, which is commonly present in the stomach and at a temperature of 37 degrees. So in the simulated environment, they created this environment where the temperature was maintained 37 degrees. Gastric fluid was provided, sodium nitrite was made available, and then again they found NDMA levels in the millions, far above the 96 nanogram limit set by the FDA. So this meant that NDMA may not be present in ranitidine tablets of any one batch. Maybe they said that all the batches have to be tested, and it is very likely to be formed once it enters a person's body. So anything could happen. Even in the stomach, this may be formed. Even before entering the stomach, it could be formed. Then subsequently, ValueSure filed a petition to the US FDA on September 9, requesting that the regulator should recall all ranitidine products from the American markets. And one of the key bits of evidence which they provided to the court was the 2016 Stanford University study. In this study, investigators gave 10 participants a tablet of ranitidine each and measured their NDMA levels in their urine for up to a day before as well as after taking the drug. And they found that NDMA levels had spiked as much as 400 times to 47,000 nanograms after the drug was taken. And the study participants were likely exposed to levels similar to those found in the ValueSure study, a few million nanograms. So the study was done by the ValueSure. Their evidence was based on the 2016 Stanford study, in which the tablets were given to volunteers and their urine levels were measured. Now, the drugs which I have enumerated in the first slide, the angiotensin receptor blockers, these hyperacidity drugs, and the antidiabetic drugs, they are not the alone which are prone to NDMA contamination. The European Directorate for Quality Medicines Management and Healthcare Management has listed many possible drugs, which are potential drugs which could be, in fact, contaminated by this impurity because of the similarities in their structure, in their manufacturing processes, etc. And they have said that all these drugs which you see in this particular slide, they must be studied, they need to be investigated, because they could be potential candidates for NDMA impurity. Among these, I can see the cefazolin, pregabalin, the trimipramine, and erythromycin, cefoxitin, which are very commonly used, sulbactam, metformin, etc. So some of the very commonly used drugs are also likely to be affected by NDMA impurities because of their structure, because of their manufacturing processes, because of the chemicals and solvents used. So, it has opened up a Pandora's box, and it has opened up the gamut of studies. We need to study those who are experts in pharmaceutical chemistry. One good thing about pharmacists is that we know pharmaceutical chemistry much, much better than doctors. Doctors do not study medicinal chemistry as much as we study. So we are the best people to rely on when it comes to the structure-activity relationship. when it comes to how the molecules are synthesized, what ring structures are there, and what can lead to tetrazole ring structure as well as NDMA formation. So the companies that have been affected by this fiasco, there have been a whole lot of national and international companies that have been affected. For example, the pharmaceutical manufacturers that were made to recall the products containing angiotensin II receptor blockers included valsartan, losartan, and irbesartan. The pharmaceutical companies were AS Medication Solutions, Aurobindo Pharma, Avcare, Hetero, Camber, Actavis, Byrant, Heritage, Legacy, Masliods, Mylan, Northwind, Newcare, Preferred Princeton, Rising, Sandoz, Solco, Teva, Torrent Pharmaceuticals. And the pharmaceutical manufacturers that were made to recall products containing ranitidine, most commonly referred to as Zantac, as manufactured by GlaxoSmithKline Beecham. They were American Health Packaging, Northwind, ANI, Glenmark, Precision Dose, Aurobindo, Lanet, Dr. Reddy's, Sanofi. As you can see, some of the most reputed drug manufacturing companies with high standards of quality, with a lot of good reputation in the market, their products were affected. And the pharmaceutical manufacturers that were made to recall the products containing metformin included Amneal, Apotex, Avcare, Bayshore, Granules India, Lupin, Mark Sands, Teva Pharmaceuticals. And whole lot of companies were made to recall. As I told you in the beginning itself, 53 drug recall notices have so far been issued, in which thousands of batches of drugs have been recalled, around 3,000 of them. 16 drug recall notices have been issued this year alone. And these are some of the companies and their products which have been made to voluntary recall. There's a difference between voluntary recall and statutory recall. Statutory recall is the one when the regulatory agency itself orders a pharmaceutical company to withdraw the drug from the market. But voluntary recall is a recall when the pharmaceutical company itself on their own recall the drug because of certain defects. So all these have been voluntary recalls. They have not been statutorily ordered by the regulatory bodies, though regulatory bodies like US FDA have made suggestions, and they have suggested to the pharmaceutical companies to withdraw them as a result of this impurity, which is a probable carcinogenic agent to the humans, which has been classified by the IARC under group 2A carcinogen. And what is the level of cancer risk to patients, we need to understand. The patients might be very eager to know. Suppose somebody is taking metformin or ranitidine on daily basis, how prone he is to, God forbid, catching this particular disease. Now, the European Medicines Agency has determined that the lifetime risk of cancer development as 1 in 5,000 patients. Out of every 5,000 patients, one patient may get it who had taken an affected valsartan medicine at the highest dose of 320 milligrams every day for 7 years. So 1 out of 5,000 may get who has taken the highest dose of valsartan for 7 years. Similarly, US FDA scientists have estimated that if 8,000 people took the highest dose of valsartan at a dose of 320 milligrams containing NDMA from the recalled batches for 4 years, there may be one additional case of cancer over their lifetime. So 1 out of 8,000 if it is taken over 4 years. That has been estimated by US FDA, while as European Medicines Agency says 1 out of 5,000 if taken for 7 years. So the US Environmental Protection Agency has found then an association between NDMA and liver toxicity. Basically, this NDMA attacks the liver cells, hepatotoxic agent, and then it damages the liver cells, causes liver cancer. But that's not the only cancer which it causes. It can cause bladder carcinoma, renal, pancreatic, intestinal, colon, and stomach cancers. So whole spectrum gamut of cancers can be caused by this particular impurity. Now, when we review the literature to find out more about this cancer risk, because people are more concerned, when we are telling them only 1 out of 5,000 or 1 out of 8,000 may get it over a period of 7 or 4 years, now they would be eager to know more about the studies which have been conducted. So a very brief review of literature in this slide is presented, that LCMS testing of 128 samples of crowdsourced metformin tablets contained highest NDMA from the tested samples and revealed 1565 nanograms per tablet. Remember, 96 nanograms is the limit, but 1565 nanograms were found per tablet in this study of 128 samples. This was conducted by Wu Qian et al. Then another which has been very commonly quoted, very frequently quoted, a Danish nationwide cohort study on 5,150 patients, which was followed for 4.6 years. It showed 104 cancer outcomes among NDMA unexposed participants and 198 among exposed participants. But they concluded that there is no evidence of dose-response relationship. So this Danish study concluded that it could not be, a dose-response relationship could not be established. Then a positive relationship was established between exposure and response for the intake of NDMA and gastric and lung cancer. Another study by Kank et al. Also observed a positive correlation between NDMA intake and colorectal cancer with a relative risk of 2.12 in a population-based cohort study of 9,985 Finnish patients. So this study included 9,985 Finnish men and women. And then NDA is also considered as one of the most potent liver carcinogen. Like these have been well established way back in 1950. They get biotransformed to ethyl diazonium ion by cytochrome P450 CYP2E1. And this ethyl diazonium ion can react with the nucleophilic sites of DNA and can form adducts which can induce cancer. So this is the scenario related to the cancer risk. Now, what is the advice for the patients? Many of our listeners would be eager to know what exactly should they do? Should they continue or discontinue? Should they substitute or replace the drug? So worldwide in 2017, about 10 million people took losartan, antihypertensive drug. Over 2.3 million took irbesartan, and around 1.8 million people took valsartan, according to the Agency for Healthcare Research and Quality. So huge number of people are using these medicines. Vast section of the population is using them. And many medical professionals have warned against unnecessary panic regarding these medicines. So they have urged that patients should not abruptly stop taking their blood pressure medicines, because that can lead to a rebound increase in blood pressure, which could put such patients at risk of experiencing a cardiovascular event. And therefore, US FDA has also advised that the patients taking metformin should continue taking it until their doctor prescribes them a replacement or a different treatment option, since it can be dangerous for patients with type 2 diabetes to abruptly stop taking metformin without talking to their healthcare professional. And then there are many other medications in the market that can be used. It's not that alternatives are not available. Good telmisartan, this azilsartan or aprosartan could be used instead of valsartan or losartan. Similarly, in case of diabetic patients, some suitable alternative as decided by the doctor could be used. So it is always better to consult the physician, consult the doctor, use any alternative. Do not stop the drug abruptly. Continue taking the drug until your doctor prescribes another one. And then, be careful about the recalls. Continue to watch the US FDA website, which puts on these recall notices quite frequently, and see which products have been recalled, which companies have been affected. Try not to use the products of those companies. Try not to use the products which have been affected. So what exactly is a drug recall? We have been using this term quite frequently. In order to explain it out for the sake of students present in this group, the drug recall means a prescription or over-the-counter drug which has been removed from the market. When a drug is removed from the market, we call it drug recall. And this could be done either on statutory basis or on the voluntary basis. And it has been, US FDA drug recalls are of three categories: class 1 recalls, class 2, and class 3. And within India also, the Central Drug Standards Control Organization has classified drug recalls into three categories. The class 1 recall is which causes most severe, adverse effects, which will lead to adverse effects or even death. And these are the most severe. Then class 2 recalls are the drugs that induce temporary or reversible health effects, whose effects are temporary or reversible. While as class 3 recalls occur when adverse effects are not likely to occur when consuming the drug or being exposed to it. Recently in 2017, the Central Drug Standards Control Organization of India has issued elaborate guidelines on drug recall and safety alerts. And they have also said that if it is a class 1, within 24 to 72 hours, the drugs have to be recalled. If it is a class 2 drug, the drugs have to be recalled within 10 days time. If it is a class 3 drug, the drugs have to be withdrawn from the market within 30 days time. So timelines have been fixed based on the severity of evidence. And then there are these market withdrawals which occur when a product has minor violation that does not require FDA legal action. We call it market withdrawal. We don't call it a recall when there is a minor violation. And then there are medical device safety alerts, which occur when there are unreasonable safety risks associated by using the product. So what are the directives issued by the European Medicines Agency? The steps that companies should take: they should evaluate possibility of nitrosamines being present in every concerned medicine within 6 months. They should prioritize evaluations starting with medicines more likely to be at the risk of containing nitrosamines. They should take into account findings from the review of CERTANs. They should immediately notify authorities of outcome of any risk evaluations. So they should also test their products of each and every batch which are at the risk of containing nitrosamines. They should immediately report detection of nitrosamines to the regulatory authorities. They should apply for necessary changes to marketing authorizations if there is any nitrosamine risk. And they should complete all the steps within 3 years, prioritizing high-risk products. These are the directives for the pharmaceutical companies. Within India, several actions have been taken by the Drugs Controller General of India. Ever since this problem has propped up, this drug contamination has been detected. In 2018, CDSCO banned the import of valsartan from the Chinese firm Zhejiang Huahai. In August 2018, Drugs Controller General of India ordered drug inspectors at port offices to inspect import consignments of valsartan's active pharmaceutical ingredient at the ports. And DCGI also urged all the state drug controllers to verify products of drug manufacturers in the context of NDMA impurity in ranitidine tablets. This happened in September 2019. But the state drug controllers raised several concerns that NDMA impurity does not find mention in the major pharmacopoeias like Indian pharmacopoeia, British pharmacopoeia, and US pharmacopoeia. Therefore, a need was felt that this genotoxic agent nitrosamine should be included in the IP monograph. And as per the directions of the Drugs Controller General of India, every company shall have to issue analytical certificate showing that NDMA content while distributing the drugs to stockists. They have to show there is no NDMA or it is within the limits, within the permissible limits. Until the manufacturing procedure is validated, the license to manufacture ranitidine by API manufacturers could be suspended if needed. It was also instructed that every manufacturing firm shall have to follow ICH Q9 guidelines very strictly, that is the quality risk management. And it is all about risk evaluation, risk communication, and risk management. So the pharmaceutical companies have to do very effective, very good risk evaluation studies, risk communication, risk management. And a member of Telangana State Pharmacy Council has even sought the Prime Minister's intervention on this particular issue. So these are the steps which have been taken by the Drugs Controller General of India. Though there is no evidence of any recalls from the Indian markets, there is no information on the DCGI website or CDSCO website showing that any drugs have been statutorily recalled. Though some, as I said, the valsartan was, stopped manufacture from the Chinese Zhenjiang Huahai company. Those companies which were receiving the raw materials from this Chinese company, they were made to change the source of the raw material and to stop the manufacturer from using that particular raw material from this company. However, there are no big recall notices issued, or there are no evidences of any big recalls from Indian markets so far. Now, so far as the detection of this nitrosamine impurity is concerned, it can be detected by HPLC, by GCMS, by LCMS techniques. Though GCMS is a destructive and hard ionization technique for a qualitative and quantitative measurement for volatile organic compounds. That is, in this technique, they use high temperature. they make it volatilize, and that high temperature can itself cause NDMA formation. So GCMS is not a very ideal technique, but LCMS is a good technique because there is no heating in LCMS, particularly liquid chromatography, high resolution mass spectroscopy. It does not generate heat, therefore generation of NDMA is unlikely. And therefore LCMS is considered to be one of the most reliable methods for detection of NDMA impurities. Though HPLC can also be used, because same is being widely used for formulation and quality control of nitrosamine impurity detection. And GCMS being a high temperature study, it was used by ValueSure, but then questions were raised that the process of detection itself might have led to the formation of NDMA, following which ValueSure conducted simulated studies using the simulated gastric environment. And in the LCMS is considered to be the most suitable because it's a hyphenated technique which separates and quantifies the components from a complex mixture with the help of a mass spectrometer. Mass spectrometer separates and detects the charged components, and LCMS is therefore a prominent technique to analyze large polar, ionic, non-volatile, and unstable organic compounds. So this could be the most reliable method for its detection. Therefore, in conclusion, I would like to give certain measures and suggestions which are necessary to be taken to curb this problem. First of all, the data regarding NDMA drug recalls within India needs to be made public by the Drugs Controller General of India. It should be available on their website. People should know how many recalls have occurred, how many drugs have been withdrawn, which are the companies affected, which are the drugs affected. Then guidelines are already there regarding drug recalls and alert these notices, but specifically guidelines need to be given, directions need to be given with regard to NDMA impurity for rigorous testing of all the batches. It is not only one batch which gets affected. Mostly many other batches get affected. And because, it could creep in from the chemicals, from the solvents, from the reagents, from the intermediaries, from anywhere. Therefore, rigorous market checking and lifting of samples for testing of drugs for NDMA impurities is equally important. Sources of these impurities need to be plugged. We need to see where from the drug is coming, the raw material is coming, because as I said, it could be introduced by the process or from the cross-contamination or from the source, using the raw material, or because of the degradation after complete formulation. Therefore, at every step, precautions need to be taken. At every step, we need to maintain sufficient precautions. Patients need to be made aware about do's and don'ts. They should not be panicked. They should not be confused with wrong information. Patients need to be given correct information, and pharmacists have a great role to play in all this, because they, being experts in medicinal chemistry, they can well guide the patients which drugs to use. And they are not authorized to tell the patients which drugs to use, but they can guide them regarding the precautions which need to be taken. Only doctors can prescribe the substitutes, and all drugs with tetrazole ring structure need to be studied for possible NDMA contamination. For example, ranitidine is inherently unstable drug. Ranitidine is one drug among all of these which is highly unstable and mostly prone to NDMA, because heat conditions, acidic conditions, change in humidity, temperature can generate NDMA from ranitidine. So one needs to be very careful while using drugs like Esaloc or Zantac, which contain ranitidine. And therefore, the clinicians also should avoid ranitidine, because we cannot determine the extent of NDMA creation that may result after manufacture because of several other myriad choices for reflux and gastromucosal. There are other choices of drugs. Famotidine is there, this cimetidine and proton pump inhibitors are there. And this NDMA has not been found in famotidine, cimetidine, omeprazole, lansoprazole, or S-omeprazole. Therefore, these drugs could be used by the doctors till this problem is controlled. And to conclude, Government of India must issue necessary guidelines on testing of all medicines for NDMA contamination, both for the drugs presently circulating in the market as well as those which are yet to be released. active pharmaceutical ingredients, that means the raw materials, as well as the inactive pharmaceutical ingredients, that means the excipients, which are prone to NDMA formation, should be tested under different environmental conditions to determine how product storage, heating, light exposure, and time since manufacture can affect NDMA incorporation. All these things need to be studied very well. So today, many students can get good ideas for their PhD, like they can study the effect of storage, light exposure, humidity, the time since manufacture upon the NDMA incorporation. And manufacturers of either API or finished ARB drug product should also test a representative sample of each lot, each batch produced. And the testing should include all the batches already distributed that have not yet been reached, their expiry date, as well as those not yet distributed. Any batch already in distribution with an NDMA level that exceeds the FDA published limit should also be recalled immediately. So these are the measures which companies, regulatory authorities, as well as the patients need to take in order to overcome this problem. So with this, I conclude my presentation, and I thank you all for your patient listening. I hope there was no hiccup in between. Any of the slides did not get stuck up in between, and I hope you enjoyed the session. Thank you very much. Over to you, Ms. Swati.
Thank you so much, sir. Kindly stop sharing the slides, sir.
Yeah, sure. I have stopped it.
Okay, sir. Well, sir, I am really fortunate to have got a chance to get so deep understanding on such an important topic today. And thank you so much, sir, to have enlightened us in this area with your vast knowledge and expertise. Sir, as I can see here, our audience has really appreciated this session today, and their deep interest has aroused certain queries. And so I request you kindly address their queries one by one as I read out them.
Sure, please go ahead.
And yes, sir. Sir, the question is that how can we minimize or tackle with this impurity individually?
All right, individually, we can, as pharmacists, if we consider that all of us are pharmacists, individually we can take part in patient counseling, patient education, and we can guide them, because patients are in a dilemma. Patients are confused, in anxiety, bewildered. They don't know what to do, whether to continue or discontinue, whether to substitute it or not substitute it. So first we can study this problem well, and then after garnering all the information about various pros and cons of this problem, we can suitably guide the patient, and we can also check whether the patient is accumulating this NDMA impurity from multiple sources. What are the maybe there's an occupational hazard also involved. For example, somebody is working in these conditions where there is more proneness to generation of NDMA. As I said, that NDMA can be generated from broiled meat, from cosmetics, from tire industry, from paint industry, and from many other sources. So people working in these industries, which I enlisted in the first few slides, they are doubly, they need to remain doubly cautious. So a pharmacist can assess the risk of such patients, like who can accumulate NDMA from multiple sources. And then if there's an elderly patient who is taking 4 or 5 medicines at a time, then we can see whether he also has a chance of exceeding the limits fixed by the US FDA or WHO. And then we can guide them, and we can tell them to consult their doctor. The doctor can then be well informed, and he can switch over to another medicine. So individually, what we can do is first to know the problem, understand the problem, then communicate, and then counsel and educate the people, and then give suggestions to the doctors. As I said, we can individually we can participate in risk evaluation, risk assessment, risk communication, and risk management, depending upon which area we are working in, whether we are working in a pharmaceutical industry, whether we are working in a hospital, whether we are working in an educational institute. We can research. For example, I'm an academician. We can do research. If you are in pharmaceutical chemistry or pharmacology, we can do research under different environmental conditions, different conditions of humidity, temperature, storage. What are the effects on NDMA? How it is nowadays, it's not a big deal to use LCMS. Many good institutions have LCMS. So we can go for detection of this impurity under different conditions and what impact it has on the overall limit. That's how we can contribute individually.
Thank you, sir. So the next question is that how much testing is needed by any regulation body to withdraw any pharmaceutical drug from the market?
Yes, European Medicines Agency has now stipulated that each and every batch of these drugs has to be tested. The drugs which have a tetrazole ring structure are most prone to this NDMA impurity, and all the drugs which have been enlisted as potential candidates by the EDQM. All these drugs, all the batches have to be tested. It is not just random sampling of 1 or 2 batches. All the batches have to be tested, those which are already in circulation, as well as those which are yet to be released. Each and every batch has to be tested. And then depending upon the statistical significance of the results obtained, we have to take a proper sample size, and then only we can come to a conclusion. As I said in one of the study, this Stanford study 2016, they used only few patients, gave the drug, then tested their urine levels for the NDMA content. But that can also be done if you want to do a qualitative study. But if you want to do a quantitative with a very good statistically significant results, you have to include a proper sample size. And however, for regulatory bodies, each and every batch has to be tested. For that, the sample size may not be important, but every batch is important. And whether it is distributed or yet to be released, it has to be tested.
Thank you, sir. So the next question is that how should a patient dispose of his ranitidine, the one he is having at home?
Yes, if the doctor has already prescribed a substitute, as I said, one should not immediately stop taking the drug, because that may lead to flare up in the blood pressure. That may lead to rebound increase in hypertension. So only when the doctor has prescribed an alternative, for ranitidine, it is used in hyperacidity. If already the drug has been replaced by the doctor, substituted by the doctor, now the question is how to dispose. There are guidelines for drug disposal given by the CDSCO, given by the Drugs Controller General of India. We should never flush the medicines in the toilets. That is what usually people do. It is, if the drugs which you have, they can be mixed with coffee, the exhaust from the coffee, the tea, whatever tea, the exhaust has been left out. You can put it, mingle it with the tea exhaust, the coffee exhaust, and then you can seal it in a sealed packet and then put it in the trash can. But you should not just simply throw it in the trash can or simply flush it in the toilet, because the drugs, when they are flushed in the toilet, it go back to the soil and they recirculate. So there's a vicious cycle. They come back in our water, they come back in our food, and that's how the plants and food materials get NDMA from. You might be wondering, nobody perhaps asked the question, how do the vegetables and food products get contaminated by NDMA? That is because when these, that's because the drug which is NDMA impurities which go into the soil, mix up with the underground water, and they get it from the water which is from the underground water. They may get it from the preservatives which we use. They may get it from the some sun-dried vegetables. For example, many in Kashmir we have a kind of a fish which is burnt fish and salted fish. So in that, it has been found way back in 1980s that it contains more than 1,000 nanograms of the NDMA. Way back in 1980s, it has been found. The smoked fish, very delicacy of Kashmir, a very highly popular delicacy in which the fish are smoked. And there is another, this dried fish, the sea fish, they are quite often salted and dried. And these salted and dried fish can also be a potential source of NDMA contamination. Though NDMA itself gets degenerated in sunlight or in UV light. When it passes through UV or sunlight, it gets degenerated. But the vegetables which are sun-dried or salted, they have a propensity of having NDMA impurities. So this proper method of disposal is mix them with coffee beans, used coffee beans, or tea, leaves, and then seal it in a pouch and put it in a dustbin. Or there are in Western countries, there are take back programs. They have a kiosk in the various localities wherein you can go and you can deposit your unused drugs. But unfortunately, in our part of the world, we don't have such facilities available wherein we can give the unused drugs back, or wherein these take back programs are there. So we have to use this method only. But there are adequate guidelines. One must go through the guidelines for proper drug disposal. And it is a vast topic. Maybe some other day we can talk for another 1 hour on proper drug disposal techniques.
Thank you so much, sir. Sir, there is one more question regarding this only, that have all ranitidine medicines have been recalled? And how do I know if the ranitidine that I take or I have at home is recalled?
Yes, actually, all these Valisure Pharma, the online pharmacy of USA, this is what they had insisted in their online petition, that all the ranitidine products should be recalled, because it is not only one batch or one product. Ranitidine itself is very prone to NDMA, and even inside the stomach, it can generate NDMA because of the acidic conditions. So, all the products of all the companies have not been recalled. Only the products, you can go to the US FDA website, and in that you can have the full list. I got the number 53 drug recall notices from that list only. There are 53 drug recall notices on the US FDA website, and you can see from the website all the products, all the companies of ranitidine which have been found to contain NDMA impurity. So for the time being, you can avoid only those companies and those products. Need not avoid all the companies and all the products, because all the companies have not been made to voluntarily recall. So, but the list is available on US FDA website, and you can inform yourself by visiting if you can email me, I can email that list also. Whoever needs the list, they can just drop their email. I'll be happy and glad to give them the full list of drugs, ranitidine or valsartan or other ARB or metformin, which have been found to contain NDMA impurity.
Thank you so much, sir. So there's one question, that are both the immediate release and extended release metformin affected?
Yes, in fact, the drugs which have been listed as containing NDMA, it doesn't matter which formulation you are using, which kind of product you are using, because the contamination occurs in the API. The contamination is there in the active pharmaceutical ingredient. So whether you use it as a normal release or extended release, whether you use it as a targeted delivery or sustained release form, it will be there. The contamination or impurity will be there, because API itself was contaminated. So, or the process was there, the defective process was there. So whichever formulation you are using, on the US FDA list, they have specifically mentioned in the table which I used. I could not include all the products. I used only a specimen product, only one of the products, but there are many more products which are available on US FDA website. On that website, they have specifically shown the strength of the drug, whether the dosage of the drug, and what release form which has been recalled. So you can see all the release forms, all the dosage forms, all the strengths of a particular drug. For example, valsartan have been recalled, or for example, metformin have been recalled. So that hardly matters which release form you are using. The drug matters, the API matters, which is contaminated in case of metformin as well as ranitidine.
Thank you so much, sir. So there is a question, that how to neutralize the NDMA toxicity in our body system, and what is preventive measure against this toxicity? What are the steps to identify the toxicity?
That's a very good question. First of all, we need to stay away from the source of contamination. That is, first of all, we need to avoid taking the drug after due consultation with the doctor. We need to avoid taking the product which has been contaminated, which has been listed on the US FDA website, which has been recalled. First of all, you have to avoid taking that drug. But somehow, if you have not been able to approach your doctor, or he has not prescribed an alternative, and you are still taking the metformin, which may contain NDMA, in that case, there are many other precautions like use of antioxidants. this natural chemoprevention. Natural chemoprevention is the method wherein we make use of natural polyphenols, we make use of natural anti-carcinogenic agents, antioxidants, because this happens as a result of mutagenicity. The ultimate damage is caused by the DNA adducts. Ultimate damage is caused by the clastogenicity, the mutagenicity. So we can use anti-mutagenic. There is one article of mine I had published several years ago, natural anti-mutagenic agents. That article is available online. So you can read that what are the various natural anti-mutagenic agents which if we consume in our daily diet, for example, green tea, polyphenols, etc., that can to some extent give some kind of protection, but that's not an ultimate protection. Ultimate protection lies in preventing the use of the product which is contaminated, avoiding the use of the product which is contaminated.
Thank you so much, sir. I even would like to request Dr. Mathur if he has any queries from his side also.
Yes, please, sure.
Thank you, ma'am. I have some questions for sir. A very good lecture, sir.
Thank you.
Yeah. So first question is, what will happen to the ranitidine that manufacturers have in storage and not currently in their storage shelves? What will happen to that ranitidine?
Sorry, can you please repeat which ranitidine are you talking about?
Yeah, the ranitidine that is that is available in the storage, but
Right.
not in store shelves.
Yes, that's why the European Medicines Agency as well as the US FDA has specifically directed all drug manufacturers that the drugs which have not yet been released into the market, they should also be tested. So they should, the drug manufacturers have to have a control on their supply chain. See, that's these are also listed in the guidelines regarding recall and drug alert notices issued by the CDSCO of India. And they have in those guidelines also they have said that there should be a very brisk mechanism for communication. The drug manufacturers should have good liaison with all the whole entire supply chain, these elements, that is the wholesaler, the retailer, the distributor, as well as the end user, and they should at any point of time be able to withdraw. For example, if there's a class 1 recall, class 1 means very severe drastic side effects could be produced, even death could be produced. So under those circumstances, the maximum timeline is only 24 to 72 hours. Within 72 hours, you have to withdraw the drug from the market. So the communication channel has to be very effective, very efficient, so that you can immediately withdraw the drug from the market. So using the same communication channels, they have to communicate to those distributors where the storage of the drug is happening, where the drugs are stored, whether in a hospital or in a warehouse or in a wholesale shop. They have to communicate to them. The drug inspectors have to come into action. They have to communicate the drug regulatory authorities. Regulatory authorities have to depute the inspectors, and inspectors have to lift samples for testing. They have to send the samples from each and every batch of such drug for testing of NDMA, and only after the NDMA is within the permissible limits, then only such batches can be released for human use. Otherwise, they should be withdrawn.
Okay, sir. Sir, and any injectable forms are also having this NDMA level?
No, so far, no, I couldn't see any of the injectables. All the drugs, these angiotensin receptor blockers are tablets. Nizatidine as well as metformin and ranitidine, all are tablet forms. I couldn't see any of the injectable forms as yet.
Okay. Sir, any LD50 values determined for this particular NDMA?
Yeah, actually, there are not LD50 values, but TD50. TD50 values are used. That is median toxic dose 50. Okay. And that is actually how they arrive at the maximum admissible limit, daily limit. The 96 nanograms per day has been achieved by using that TD50 dosage formula, which has been put forth by the Rogotowski. And in that, what they do is median toxic TD50 is the dose which is toxic to 50% of the population. Median toxic dose 50. So suppose the incidence of this is 1 in 1 lakh. So you have to divide 1 lakh by 50%. That is 50,000. So now the annual, if the daily intake is in case of NDMA, it was found to be 1.96 nanograms per day. So 1.96 divided by 50,000, that is what gives the 0.096 nanograms.
Okay.
And that is how they have arrived at this 96 nanograms per day.
Okay.
So these median toxic dose 50 values are used to calculate the average daily intake for every drug. Either the 26.5 nanograms is the daily permissible limit, or 96 nanograms is the daily permissible limit, which I have shown in the table. For which one there is 26 and for which one there is 96. That is actually based on the median toxic dose 50. But in there are values. If you want to do the study on animals, those LD50 values can also be found out, and you can conduct the study on animals using those LD50 values, because already the human dose has been specified. We can calculate animal dose also.
Okay, sir. Sir, there are 3 questions more.
Yes, please go ahead.
One question is that is this NDMA level gets affected by the temperature and pH variations?
Yes, of course. Yes, of course. It gets affected, as I said, when the detection GCMS was used as a detection method, the temperatures rise to 180 degrees.
No, sir. I am talking about this particular storage area. Suppose if any drug is stored there.
In storage, the temperature, the humidity, the time lag from the date of manufacture, that can also affect how long it has been in storage. What was the temperature condition? What were the humidity conditions inside the storage? All of them can affect, because it is the ranitidine in particular, that is very highly temperature specific. If the temperatures are rising, for example, in many parts of India, we have during summers 45 to 45 degrees temperature. And if drugs are stored in those conditions for a long period, there is every chance that NDMA may be more. And the Valley Shore, they found millions of nanograms per tablet. The limit is only 96 nanograms. So maybe, yes, temperature, humidity, storage conditions are very, very important.
Okay. Sir, how a person, if he is taking metmorphin, he is sure that this metmorphin is not having ranitidine, or how he can confirm that his metmorphin is not having ranitidine?
Yeah, if he can, of course, a common patient, a common consumer cannot test the drug on his own. He doesn't have the wherewithal, the means, the equipment, LCMS, the knowledge or the expertise to do that. So best a person can do is to go back to the US FDA website, check the list of drugs which have been recalled. There are 53 drug recall notices. So all the drugs are listed there, all the batches, all the strengths, all the formulations. So he can see if this particular drug belonging to this particular company of this strength and formulation has been affected or not. If it is there, he can consult his doctor. He can get it replaced by some other drug, and he can immediately, no need to stop it immediately, but after consulting the doctor, he can stop taking the drug. I think that is the only means by which a patient will get to know, by referring to the US FDA website. There's a full list available, or by consulting his pharmacist or by consulting his doctor. Pharmacists are expected to know all this. So pharmacists can also guide them, doctor can also guide them. But of course, common patients cannot test the drugs on their own.
So last question from my side. That as you told that the food items are also affected by NDMA levels. Okay.
Yes.
So is the water also affected by NDMA level?
Yes. Water is affected.
And
air is
what's
affected.
the source in it?
Water is affected. Air is affected. Cosmetics are affected. There are limits for everything. There are different limits. As I said, for medicines, the maximum daily admissible intake is 96, but WHO allows up to 180 for water. So for water, the limit is higher. So that is 180 nanograms per day. So I think it is 180 may need to be checked once. So, but in any case, it is higher than the limit prescribed for the medicines, because medicines we are taking on daily basis, and medicines we are consuming for a long period. Of course, water also is consumed for a long period, but there are certain things which we cannot avoid, but we need to be certain that overall intake from the food, from the water, as well as from the medicines should not exceed the daily permissible limit. If it is exceeding the daily permissible limit, then we are at the verge of we are exposed. So that needs to that's where again we need to test the urinary. There is one field which is emerging these days, that is pharmacometabonomics. That is, we take the metabolites of a person and we find out the concentration of various toxic substances or the concentration of various medicinally active substances. I have presented a paper on this particular topic in an international conference. So by using these pharmacometabonomic techniques, we can find out the metabolites from the body fluids. We can find out we know the metabolites of nitrosamines. So we can detect them from urine, we can detect them from other body fluids, and we can see if the plasma concentration of these nitrosamines is within the permissible limits or not. If somebody is exposed to the occupational hazard, somebody is living in bad conditions wherein the water is likely to be polluted or the food items are likely to be contaminated, then I would suggest that he should go for these pharmacometabonomic studies, and he should get his body fluid samples tested to see what is the level, plasma level or urine level of these NDMA metabolites. That will be an ideal thing to do.
Okay, so one only last associated question with this.
Yes, please.
How a person can determine if suppose NDMA level is affecting him? If any clinical signs or symptoms?
Yeah, for that, we need to go back to the pathophysiology. We need to go back to the, this diagnosis of diseases. If, some people, first of all, the US FDA itself has said 1 in 8,000 persons gets it after 4 years of consumption, or 1 in 5,000 persons gets it after 7 years of consumption. So even if a person continues to take, for example, now somebody gets to know that his drug is affected, the drug which he has been taking has been shortlisted, has been recalled. So even if he continues to take the drug for another 1 month or 2 months, will not expose to him to a potential carcinogenic risk, because only after several years of usage this risk gets accumulated, and on short-term use it has not been found to be highly risky. So short-term use can be continued until he consults his doctor. But how will he get to know? He will get to know if somebody has been taking it for several years and he suspects that maybe he might have been affected. Then he has to, go look at those signs and symptoms. For example, in case of, the general common symptoms of cancer, breast cancer, lung cancer, we know there is coughing and there is pain, or there is any, abnormal patterns of eating or abnormal patterns of sleep, or there are any outgrowths on the body, or there are, while eating, somebody is not able to swallow the food stuff, or his bowel habits have suddenly changed. Yes. There has been suddenly a gain in the weight or loss in the weight. Sudden gain, body weight gain, sudden loss in the body weight, or bowel habits have changed drastically, or sleep patterns have changed, or the outgrowths have increased on the body, or allergic conditions are manifesting differently. Anything abnormal. See, it is even for the normal people who do not suspect to take any NDMA. Even they need to be very cautious about these things. Certain changes, certain abnormal, unexpected changes taking part in our body, they need to be taken very seriously. We need to consult a doctor who would prescribe certain diagnostic tests, and only after undergoing those diagnostic tests, we'll get to know whether we are safe or not. So any abnormal symptoms and unexpected, unusual symptoms, changes in the body, we should be very careful about, we should be very keen about, and that should be taken to the doctor and reported to the doctor.
Thank you so much, sir. It was indeed a great lecture.
Thank you.
So I again have certain questions, if you don't mind.
Please, not at all.
So the question is that how many times should a manufacturer test the presence or limits of NDMA in drug, as the drug like ranitidine has 3 years expiry?
Yes, the regulatory authorities have necessitated testing only once. They have not said repeated testing is necessary. They have, but if the drug is being stored for a prolonged period under abnormal conditions of temperature or humidity, if the drug is being stored under normal conditions of temperature and humidity, maybe repeat testing may not be needed. But if the temperature conditions or transportation conditions are not normal, they are not as as per the guidelines, then probably we may otherwise also, when we manufacture any pharmaceutical product, we do conduct this in-process testing as well as end product testing. Then we also conduct testing before transportation as well as after transportation. That is at the recipients, and also the testing is specified for any pharmaceutical product. When you are purchasing for a hospital, for example, the hospital has to test each and every batch of the drug which they have received. Quality, efficacy, and safety. These are the three important parameters for which any drug has to be tested, whether by the pharmaceutical company manufacturer or by the hospital. And for this particular reason, the NDMA, if the drug has been stored for long, it has been quite long since its manufacture. Now, for example, 1 year or 2 years since manufacture, I would suggest one should go for testing, repeat testing. But regulatory authorities have not specified any such thing so far, or maybe I don't know about that. So I didn't see any specific guidelines regarding repeat testing by regulatory authorities, except that they have said that each and every batch has to be tested, and before and after shipment they have to be tested. But repeat testing, if prolonged storage is suspected, if abnormal temperature, humidity conditions are suspected, then obviously one should go for repeat testing also.
Thank you so much, sir. So just one last question to wrap up the things. If a patient is having some heart ailment and he's already taking angiotensins and anti-diabetic drug, so how to detect that how much NDMA has been consumed in the body and how to prevent it?
Yeah, I would say once again, if a patient suspects that he has been taking a drug which was recalled, which was labeled, which was found to have NDMA, then that patient should probably get his body fluids tested for NDMA levels. He should get his pharmacometabolomic analysis done, so that he gets doubly sure that he has not been exposed to very high levels of NDMA. Otherwise, once the testing is done and there is no such thing, no such risk that his body fluids are not showing any abnormal levels of these NDMA or its metabolites, then there is nothing to worry. For short term, they can continue to use the drug, but consult the doctor and change them. And they, if they have to see that what are the symptoms which they have been observing. The symptoms may, or as you said, for example, a patient may be a heart patient. So whatever symptoms he may be having may not be, the NDMA does not produce any effects on heart per se. It does not produce any cardiovascular abnormalities, but it is believed to produce only teratogenic insults. These mutagenic insults, and it is believed to produce the carcinogenicity. So he need not suspect that it could be because of NDMA. NDMA would only cause carcinogenicity after long-term usage, that too when a person is taking a drug which is far exceeding the permissible limits. Otherwise, on short-term usage within the limits, there is nothing to worry.
Thank you so much, sir, for quenching our thirst of knowledge to this extent. And thank you for answering all the questions with so much interest and passion.
Thank you very much.
Thank you to all our viewers to have stayed over so patiently and have enthusiastically participated in this session today. We are really gratified by your presence today. Thank you so much, sir.
Thank you very much for this very interactive and interesting session. It has been equally enlightening and enriching for me to listen to the number of questions. That has provoked a lot of thinking in my mind also. After going home, I would also be provoked and prompted to study more on this subject, looking at the number of questions which have been raised. So of course, it is a mutual benefit to both the speaker as well as the listener. Highly thankful to Dr. Shashi Alok ji, Dr. Abhishek Mathur ji, yourself and everybody, all the listeners, all the members of the audience. Thank you very much once again.
Thank you so much, sir. We are really honored to have had your presence today.
Thank you.
Pleasure.
Thank you so much.